Hello and welcome to another episode of the Genepool series. For this episode, we'll be talking about the changes in genetic diversity from 1886 to 2001 using 96 Canadian oat cultivars that are analyzed for genetic diversity. This paper is written by Yang Fu in its goal of quantifying how the gene pool has changed over time. Let's get started. One of the primary concerns about plant breeding has always been the reduction of the total crop genetic diversity, which diminishes the amount of variability in disease resistance and environmental tolerances. This limits the ability for crops to respond to new agricultural practices, changes in the climate, and pests and disease. In the UK, there's been a 35% reduction in the gene pool in winter wheat. Oat breeding in Canada began in the late 1800s to meet the demand for growing Canadian livestock. Improved methods of hybridization and selection occurred from the 1900s to the 1930s. This occurred at Agriculture and Agri-Food Canada, or the AAFC, which is a research branch of the Canadian government that specializes in agricultural research and plant breeding. Several cultivars were bred within this organization, in conjunction with several Canadian agricultural colleges that existed during that time. In the 1960s, two highly productive cultivars called Liberty and Legacy were developed. These were developed through backcrossing rest-resistant genes into the cultivars Rodney and Pendick. Dumont and Robert are also examples of such cultivars. The use of wild oats as a breeding parent in the 1970s further enhanced the development of many different cultivars, with resistance to both rust diseases such as stem rust caused by Puccia graminis and crown rust caused by Puccia cornata. So far, the breeding program has produced as many as 130 registered cultivars, many of which made a major splash within Western Canada, and are the primary cultivars grown in Canada. In spite of this success within breeding for improved yield and resistance, there's a heavy concern about the narrowing of the gene pool of Canadian oats. Since 1930, the gene pool has largely been based off of 10 parental lines that act as a foundation stock from which all of our modern cultivars are derived. This is similar in the United States where all cultivars before 1970 could be traced back to only 7 lineages, obtained through introduction from Europe. Of the 130 cultivars released in total, 96 were selected to be studied. These represent the most commonly used cultivars throughout that history. These were analyzed using the SSR genetic marker technology to determine how genetically distinct these individual cultivars are. And here are the results. Through this genetic testing, it was found that the average increase in the gene pool declined from the 1930s to now. It started off at an average increase in genetic diversity of 1.29 in the 30s before declining to 1.27 to 1.27 in the 40s, down to 0.92 in the 50s, down to 0.6 in the 60s, down to 0.53 in the 70s, up to 0.62 in the 80s, and up to 0.64 in the 90s. So there is an effort to reverse courses of the gradual decline in the gene pool. The increase in the gene pool and decrease in the gene pool can be seen on screen right now through different measurements. This research project discovered that there has indeed been a negative impact from the oat breeding management strategy throughout the history of the breeding program. This means in the long run, future improvements will be inhibited by this lack of diversity going forward. The early improvements in genetic diversity occurred as a result of the early use of hybridization in the 1920s and 1940s, even though there was somewhat of a line in the gene pool overall, due to the limitations of how many genes were added into the mix each subsequent generation. The act of hybridization, instead of just pure selection, broadened the gene pool from the perspective of modern cultivars. leading to improvements in oat yields, improved disease resistance, and making the gene pool wider. From the 1950s onward, the attempt to combine all the existing rust resistance genes together led to hybridizations between elite cultivars through backcrossing and breeding elite with elite. This would over time reduce total genetic diversity, and a similar phenomenon has happened both in corn and barley. This means that effective selection is needed to make sure that the gene pool remains wide in order to prevent any further decline in the gene pool. The researcher of this paper recommends the use of SSR genetic markers in order to assess each individual cultivar's genetic variation and similarity to other cultivars, as a means of making sure that the gene pool remains wider and continues to widen over time. The researcher also recommends the use of more novel species and cultivars to be introduced into the overall gene pool through the process of integration and hybridization. And that about covers everything. Thank you for watching. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.